Assalamu alaikum. Welcome student in our biology online class. Today we will discuss about chapter number 6, enzymes, and the topic from this chapter is characteristics of enzymes. In this chapter, characteristics of enzyme, if we talk about first point, is that protein nature of enzymes. Almost all enzymes are protein. They are made up of amino acid. Now this means that enzymes are protein in nature, and the basic unit of enzymes is amino acid. Now student, two or more than two amino acids combines to form various kinds of protein. Now here is a diagram of amino acid in which a central carbon atom is bound together by amino group, hydrocarbon, acyl group, and carboxyl acid group. All these are bind together and form amino group. Now the next one is discovery of enzyme. Enzymes were discovered by Wilhelm Conhe in 1877. And according to him, the enzymes are globular protein. Now when a long linear chain of amino acid folded together, then they form a three-dimensional structure of amino acid. Now in this picture, look at this picture. In this picture, a long chain of protein is specific, now which is known as globular protein, and these globular proteins are enzymes. Now the next one is characteristics of enzyme is increase the rate of reaction. The rate of those reaction which are catalyzed by enzymes are millions of times faster than those comparable uncatalyzed reaction. Now those chemical reaction in which the enzyme uses is millions times faster as compared to those reaction which are without enzymes. Yes, I understand. Now there is a diagram. You can easily understand that enzymes and without enzymes, right? Here is this picture, the with enzymes or without enzymes. Now in any chemical reaction, enzymes less the requirement of activation energy, or in a very short time the reaction speedily and the substrate will convert into product. While if the chemical reaction is without enzyme, now look at the picture, the without enzyme, you see the activation energy is more, right? It takes more time to convert reactant into product because of the high demands of activation energy. Clear, student? Now the active site. Only a small portion of enzyme molecule is directly involved in catalysis. Now this region is known as active site. It recognizes and binds substrate and then carries out the reaction. Now student, enzymes in any chemical reaction cannot participate as whole. Okay? Instead, only a small portion of enzyme. Understand it? Only a small portion, which we called as active site, and that only participate in the chemical reaction. The active site of the enzyme is recognized the substrate and bind with the active site of enzyme. Now here is enzymes and the enzymatic reaction. Here you can clearly see the active site and how the substrate is attached with them. The whole enzyme is not involved in chemical reaction. Only here is the active site. Here is the active site on which the enzyme react. Clear? Now this active site bind the substrate and the substrate converted into product. Now these enzymes help the substrate to convert into product. So the active site is only that small portion of enzyme that is utilized in a chemical reaction. Clear? Now, not consumed by reaction. This is the next point. This is the next characteristics of enzyme. Means enzymes are not consumed by the reaction they catalyze. Means enzymes are not consumed by reaction or they catalyze. If enzyme catalyze any reaction so that substrate convert into product and releases by themselves, means enzyme in the reaction. Look at the picture. In this picture, enzyme bind the substrate and that substrate convert into product, and in the end of the reaction they are present as such in the original portion. Yes, look at the diagram. In this diagram you are clearly seen that the enzymes, this enzyme attached that substrate and converted the product, and in the end these enzyme is as it is at the starting of the reaction. Clear, student? Now as you know that catalyze the chemical reaction means that enzymes are not wholly consumed. Understand it? Now the next one is enzymes are specific. Enzymes are specific in nature, means enzymes are very specific for the type of reaction, for the nature of the substrate. Here, any specific type of enzyme is for any specific type of substrate to convert into product, and the specificity of enzyme is depend upon that which type of substrate it is. Now that substrate is converted into product, an enzyme attached that substrate which is specific for that enzyme. And what type of chemical reaction? So according to that type of specificity of enzyme is attached to the substrate and convert into product. Now you can easily understand by this diagram. Now in this diagram, a variety of substrate and enzyme can be seen. Now for lactose, yes, look at this. For lactose, there is an enzyme, lactase. Clear? And for glucose, the enzyme, galactase. And for the nature of enzyme depends upon the type of substrate. Now here is another substrate, sucrose, and the specific lactase is an enzyme is produced which can convert this sucrose into a product. Now here is enzymes which are specific for the type of reaction and for the nature of substrate. Hope you understand this. Now here is some enzymes and their function and the origin where these enzymes are produced. Now here is number one, salivary and amylase is an enzyme, and the origin of that enzyme is salivary gland. And from where these salivary gland? These salivary gland produce in mouth. So the salivary gland is only produced in mouth, not the other place, not the other part of the organ is produced that type of enzyme. And amylase is converted into maltose. Here is the only that enzyme is attached, starch and glycogen is a substrate over here, and that amylase is only the enzyme that converts into product. Clear? And that is the place where mouth. Now here are some other enzymes where the places which are produced and the substrate which are attached with them and the product which these enzymes converted. And here are the origin where these enzymes are used. Clear, student? Now the enzyme's activity can be regulated. Now according to the need of the cell, the activity of enzymes can be regulated in inhibitor and activator. Now here, what is the meaning of activator? So activator are those who enhanced or speed up the activity of enzyme, while inhibitors are those who decrease or stops the activity of enzyme. Now enzymes production can be enhanced or diminished according to the requirement. Now here, according to the need or the requirement of the cell, the production of enzymes can be increased or stopped. Now let us understand by this diagram. In this diagram, if any specific cell need a specific type of product, and in that time the active site of enzyme is available or free, so substrate get attached with the active site of that enzyme and converted into product. But at some time, if cell does not need that type of product, here is the meaning of the cell. Matlab, if cell needs a production, then the substrate attached with enzyme and converted into product. Or if the product stop, the production of that cell will stop the substrate into convert into product. Then there are some inhibitors. And that inhibitors attached or bind the active site of enzyme, and that enzyme cannot bind substrate to enzyme or inhibit the enzyme to attach that substrate, and that substrate cannot bind the active site of enzyme, and in this way inhibit the active site of enzyme to attach the substrate. So production of enzymes in the cell, inhibitor uses according to the requirement of cell, inhibits or uses activator to activate the enzymes. Clear? Now student, the next characteristics of enzyme is cofactor. Some enzymes require non-protein molecule or ions as additional component to work called cofactor. Means performing the activity of enzyme, there are some cofactor which required and they are non-protein in nature. Now there are two types of cofactor: inorganic, metalline, and organic, flavin or heme. Now inorganic can be metallic like zinc, magnesium, copper or iron ion, while organic cofactor are mostly vitamin like flavin or heme. So that type of cofactor help enzyme to work out. Now here is a diagram in which it is a protein of enzyme while it is a non-protein cofactor, which is non-protein in fact nature. They bind with the protein portion of enzyme to coordinate or help out the enzymes for their working. Types of cofactor. Here two types of cofactor. Prostatic group. In this group, organic cofactors are highly bound to enzymes and coenzymes. Coenzymes, in this group, organic cofactors are loosely attached with enzyme. They transport chemical group from one enzyme to another. Now, what are prostatic group? In this group, organic cofactor are tightly bound to enzyme, while in coenzymes, the organic cofactor are loosely bind or attached with enzymes and they transport chemical group from one enzyme to another. The examples are riboflavin, thymine, folic acid. Now here is a diagram. In this diagram, cofactor tightly bind with protein portion of enzyme. So we can say that this is the prostatic group. Or if that organic cofactor is attached with protein part of enzymes loosely bound, then this will say that is coenzymes. And this coenzymes loosely bound. Now the cofactor mostly used in that type of chemical reaction where these coenzymes require transfer one enzyme to another enzymes. Means the requirement of enzymes from one enzyme to another enzymes. This organic coenzymes are mostly the vitamins, and the vitamins like riboflavin, thymine and the folic acid. Let's talk about the next one. Enzymes can work together in a specific order. Now in this characteristics of enzyme, that enzymes can be worked together in a specific manner. Now several enzymes can work together in a specific order. and that order is creating in the metabolic pathway. One enzyme take the product of the second enzyme as a substrate, and after the reaction the product onto the next enzyme. Now you can understand this. Sometimes some enzymes work out in this one enzyme product is attached to another enzyme and used as a substrate, right? Now here is a diagram. Here is a diagram. You will understand how the enzymes is as a substrate converted your product into second enzyme and then into third enzyme. Now the substrate, one enzyme substrate is product in a product, and that product is converted to next enzyme as a substrate. Now this substrate is transferred from as a from one enzyme product is transferred to second enzyme as a substrate. Now for example, in living organisms there are many metabolic pathway like respiration, digestion of food and photosynthesis, etc. In these metabolic pathway, the several enzymes work together in a sequential manner to produce a specific product, and this pathway is called metabolic pathway. Now here is some enzymes which work out in a sequential manner and produce a product. Clear? Hope you understand the characteristics of enzymes clearly. Thank you, student.